Welcome, Dr. Bjorkman.
Thanks very much. Thanks for having me. And we still work on HIV, but I thought I'd share the coronavirus research we're doing. I think everyone's aware that it's a lot easier to make a vaccine against SARS-CoV-2 than it has been against HIV, but we haven't given up on that. It's just that we haven't been able to make one like anyone else yet. Okay, so I wanted to talk about the COVID-19 work we started doing at the beginning of the pandemic. And so just to bring everyone up to speed, I thought I would, oh, my slides won't advance. That's interesting. Okay, sorry about this. Okay, all right, so SARS-CoV-2 is a coronavirus, and like lots of other viruses, including HIV, human coronaviruses emerged in animal hosts. And so we've had, over the past 20 years, we've had 3 epidemics or pandemics. So the first was SARS, the original SARS, that I'll call SARS-1. So that was in about 2003. Then we had MERS, which was in 2012. And they were epidemics rather than pandemics because they kind of fizzled out. And then now we are in the middle of SARS-CoV-2. There are 4 common cold coronaviruses that have been with humans for millennia, really, and don't cause serious disease most of the time. So let's get to SARS-CoV-2. It's an envelope virus. So like HIV, it has a membrane surrounding its nucleic acid, which is RNA in this case. And it uses its spike protein, which is a counterpart of HIV ONV. It's also a trimer, to recognize its receptor, which is ACE2, which lines epithelial cells in airways, and also it's on the surface of other organs. And so like other viruses, basically to simplify it, coronaviruses need to enter the host cell, make copies of itself, and release new progeny viruses from the cell to infect other cells in the same person, and then go on and infect other hosts. And so the interaction that's important for that is the spike trimer interacting with ACE2, and that triggers changes that allow the membrane of the coronavirus, of the SARS-2, to fuse with the host cell membrane. And neutralizing antibodies, of course, can prevent that. It can prevent viruses from infecting cells, so then they can't make new viruses. So the interesting thing to me about coronaviruses, and this applies to all of them that I've seen the data for this, is that they do not actually hide their receptor binding domains from antibodies. So a lot of envelope viruses that have fusion machinery, glycoproteins, manage to try to hide their receptor binding portions from the immune system so that antibodies can't easily neutralize them. And what I think is really unique about coronavirus spike structures, as I said before, they are a trimer, but they have these receptor binding domains. So they have 3 of them because there's 3 per trimer. And those can be in either what's called an up position, or over here on the left, a down position. And so you can have 3 up and 0 down, or 2 up and 1 down, and so on. And the interesting thing about that is when the receptor binding domain is up, it is exposing epitopes that the immune system can go after and make neutralizing antibodies against. And I'll come back to that at the very end of my talk about our Mosaic vaccine approach. Okay, so we started on this when Christopher Barnes, who was then a postdoc in my lab, started solving cryo-EM structures of SARS-CoV-2 spike trimers bound to neutralizing antibodies that were isolated from COVID-19 donors by Michelle Nussenswag's lab. So they had this early paper by Davide Robiani that was in Nature, and they had a bunch of antibodies that they'd isolated from COVID-19 donors that they collected from in like March of 2020. And so Christopher did a bunch of these structures, and we came to some general principles that I'll talk about in a moment. But they all recognized the RBD, which is the majority of neutralizing antibodies are against that receptor binding domain, or RBD. So what you're seeing here are The spike trimer structures in light gray is the S1 subunit that has the RBDs on it, and in dark gray is the S2 subunit that's got the fusion machinery. So it's kind of like the counterpart between GP120 and GP41 in HIV on trimer. Okay, so the colored things here are the variable regions of different antibodies bound. And so you can see that there's all kinds of ways the antibodies recognize the spike trimer. They can recognize either down RBDs. This one here, C144, has to recognize down RBDs, but others can recognize both up and down RBDs in different ways. And so we had a lot of these structures, probably 40 or 50 of them. I'm only showing you a few, but there were a lot of different principles that came out of these studies. And then after this, Christopher did more structures from antibodies that were isolated from people who'd received one of the mRNA vaccines. And that was a study led by Vinci Wong in Michelle Newson-Swog's lab, and Christopher did a bunch more structures. And basically the antibodies they're all slightly different from each other, but they obeyed the same general principles. So what we did from there was to classify the RBD binding antibodies into 4 classes based on what their epitopes are on the RBD, where they are, and then on the properties they exhibit when they're bound to the spike trimer. So what we've done here is we've taken computationally, we've taken away the RBD from a spike trimer structure, and then we use information from the structures that are always on the trimers. Okay, so people have a lot of different classification systems. This is just the one that my lab uses. So what we called class 1 antibodies were antibodies that block ACE2 binding. They actually sterically overlap with the ACE2 binding site on the RBD, and they can only access the RBD if it's in an up position on the trimer. Class 2 also overlaps with the ACE2 binding footprint, but they can bind to spike trimers if the RBDs are either up or down. Class 3 were sort of lower down on the RBD, as shown in this depiction. This is like one particular antibody. One example antibody is shown in each case. And one example of this antibody is S309, which was an antibody from the company Vir called sotrovimab. And up until recently, it was used therapeutically, but it no longer recognizes Omicron variants. Okay, and then there was class 4, which early on were a couple of antibodies that were binding to a part of the RBD that's a bit hard to access when it's associated with the spike trimer. And I'll come back more to this in a moment. So the original antibody in this class was something called CR3022. It was actually isolated from a SARS-1 infected patient a long time ago, and it cross-reacted with SARS-CoV-2, but it wasn't very neutralizing. Okay, so what we're going to be talking about here is the fact that there's another kind of subclass here, which we call class 1/4, and it's in between class 1 and class 4, and it's not as hard to access. And the important thing is that this part of the RBD tends to be more conserved than the top part. So what I'm going to show you now is the structure of the spike trimer with a gradient that we're going to be using throughout the talk, where we plot the variability among SARS-like beta coronaviruses. So SARS-1 and SARS-2 are both Sarbecoviruses, which means SARS-like beta coronavirus. And so we're going to plot the sequences onto the structure of the spike trimer, and we're going to go from orange, which is variable in the amino acid positions, to purple, which is more conserved. So in this movie now, we're going to start with the spike trimer structure as a surface representation. You see S1 and S2, those are the RBDs. And so in yellow, they're in yellow in a cartoon representation, that's down, that orange one, but now it's in an up position. And now we've plotted this orange and purple gradient. So the purple part is more conserved, and the class 1/4 epitope is actually quite conserved compared to the top of the RBD, which is the class 1 and class 2 epitopes. So there are examples. We found C118 and C022, which were monoclonal antibodies isolated from those original COVID-19 patients early in the pandemic. And so they don't neutralize quite as well as the ones that they were concentrating on first. Those are the ones up here at the top. But if you remember, the top is the class 1 and class 2 epitopes that are less conserved. And so the problem with the class 1 and class 2 epitopes is that they keep changing in all the variants. And so the ones isolated early on no longer work. So there's been a series of therapeutic antibodies that were against that class 1 or class 2 region, and they no longer work. Okay, so what we wanted to do was actually said it in the movie. We wanted to make we wanted to target the base of the RBD, which are the class 3, but mostly the class 4 anti-RBD antibody epitopes. We wanted to target those because we wanted to make a vaccine that would then not be subject to, no longer working. So originally we were trying to target animal Sarbecoviruses that could emerge during another spillover event, and that's still what we're trying to do. But then later, all these variants of concern arose, and those tend to vary in the same orange positions. Okay, so now if I go on to, I won't play the movie again, I think. Okay, so how are we trying to preferentially target the conserved region? So this is schematically the theory behind what we decided to do. So the idea is we make these nanoparticles that multimerize an antigen. So we're doing this for HIV as well. So basically there's a protein-based nanoparticle to which we attach an antigen. So in this case, it's an RBD. So now we can schematize the RBD as having orange regions, which are not very conserved, and then a purple region that's a bit less accessible in the inside. And so what will happen is if we make these nanoparticles and we've clustered the antigens, because of basic immunology, you get a stronger immune response if you multimerize an antigen. So we're doing that. But what's going to happen most of the time is when it goes into a germinal center, there'll be B cells that have membrane-bound B cell receptors that will be able to cross-link. That is, they can use both of their Fab arms to bind to adjacent strain-specific epitopes. So these are represented by the orange triangles here. So that would be like the class 1 and class 2 epitopes on the RBD. But what if instead of making this nanoparticle that we'll call homotypic, that is, every antigen is identical, what if instead we make a mosaic nanoparticle where it's unlikely that any two RBDs that are next to each other on the nanoparticle would be the same? That means they'll have different distracting epitopes. And these orange types of homotypic receptors won't be able to cross-link, so they won't have a competitive advantage, and so you won't tend to activate B cells with receptors that see the distracting epitopes, is the theory. And instead, what we were hoping would happen is that the purple B cell receptors, which are binding to conserved epitopes here, that might be a little bit less accessible, would reach between the adjacent RBDs, and the purple regions are pretty much the same. And then this B cell receptor would, in theory, have a competitive advantage. So we'd stimulate these purple types of B cell receptors. And so again, what we're trying to do is target the purple region, and on the RBD, if it's in an up position, you can see the epitope we're trying to get. It's either a class 4 epitope down here, but those aren't as neutralizing. And so what we'd really like to get is these class 1/4 epitopes, because in this paper here, Claudia Jett, together with Christopher, we reported from structural studies and competition studies that the class 1/4 antibodies, they bind to a conserved region, but they also sterically block ACE2 from binding. It's not because they overlap with the binding site, but just sterically they block. And so those are better neutralizers. Okay, and then I want to emphasize here that the variants of concern, we've plotted here Sarbecoviruses from bats mostly, the sequence variation, and it's all, the variability is mostly in this class 1 and class 2 region. And we've plotted a bunch of the variants in the variants of concern, including all the Omicrons, and you see the same orange and purple distribution. So it doesn't matter if you plot it from Sarbecoviruses or the SARS-CoV-2 variants of concern. Okay, so how do we actually make these mosaic nanoparticles? Well, we've been using the system Mark Howarth developed when he was at Oxford. So basically there's a bacterial protein he calls spycatcher, and that protein will bind covalently to anything that has a spy tag on it. And the spy tag is just 13 to 17 residues. It's a piece of the original protein, and he removed it, and now you just add 17 residues or so to, let's say, your RVD. You mix it with a nanoparticle, and I'll show you what that looks like. It has 60 spy catchers on it. They've fused spy catcher to a protein-based nanoparticle. And so now you can make what the yellow ones are homotypic nanoparticles very easily by just incubating with one type of RBD. And you can also make mosaic nanoparticles by incubating with 2 RBDs or 3 RBDs, or as I'll show you, 8 RBDs. So again, that's what we spy tagged the RBDs. So we made 8 different spy tagged RBDs, and they all coupled to spy catcher nanoparticles, the same efficiency. And that's a computer model of what it looks like. It's a nanoparticle called MI3, which was based on one of Neil King's. He's at University of Washington. It's based on one of his designed nanoparticles, and it has 60 places to which the Howarth lab attached spy catcher. So if you can see this, the colored things are different RBDs arranged randomly here. And then I don't know if you can see it, but in dark blue is the spy catcher attached to MI3. Okay, so this started in 2021 when Alex Cohen was then a graduate student in my lab. He's now a postdoc in my lab. But what he did was early on to choose RBDs from 8 Sarbecovirus spike proteins to make these mosaic nanoparticles. And he picked Sarbecovirus strains from a paper from Vincent Munster's lab by Michael Letko. It's this paper right here. And so they were talking about which Sarbecoviruses had a potential to spill over. And so we included, originally we included the original strain of SARS-2. Later we updated it to put beta on. But as I'll show you, there's really no point in our vaccine strategy to update it again. Like no point in, we don't need to keep updating. But anyway, we stopped at beta. And then we added these bat RBDs, and one of them's from a pangolin. Okay, so we chose these because they had spillover potential. And then we deliberately left off strains that we wanted to use for testing. And the most important one was the original SARS. We left that off because we wanted to use that in a challenge. So what we're going to be doing is looking for, we inject animals, and then we looked for immune responses to so-called matched strains. And that means, you know, the strains listed right here where they had a RBD on the particle versus mismatched strains where they do not have an RBD on the particle. And then, of course, as the pandemic went on, we added more and more and more mismatched strains as the Omicrons built up. And, you know, we're working on getting, I mean, we're out of date here because this was from our paper in July, and now we haven't yet added in neutralization. Well, we have neutralization capability now for some of the new Omicrons. I think we're in the middle of making XBB pseudoviruses. Okay, but the point is we were looking for matched versus mismatched strains. And very importantly, we kept, there's all these different nomenclatures and people who classify these into clades. And the RBDs on the particle are clades 1A, 1B, and 2. And we deliberately left off these clade 3 Sarbecovirus RBDs because those are more distantly related, and we wanted a test of how far away we could go in terms of getting mismatched responses. So we were always most interested in the responses to the clade 3 RBDs. Those are more different than the ones on the particles than the Omicrons or other variants of concerns are. Okay, so this is from a paper that we published in July where this is the title of that paper. So we wanted to do challenges in animal models. And so what I'm going to tell you about very briefly are that we did an experiment where we compared injection of Mosaic 8 nanoparticles right here versus homotypic nanoparticles using, this has got 60 copies of SARS-2 beta RBD. This has 60 RBDs, but they're 8 different ones. We immunized, I'll talk about this later, a transgenic mouse that expresses human ACE2 receptor. We looked for binding and neutralizing antibody responses. So by ELISA binding assays and neutralization responses. And then we did challenges against SARS-CoV-2, which is a match strain, versus SARS-1, which is a mismatch strain. And then we also did a challenge study in non-human primates. This was done at BioQual, a contract research organization. I should say this was done at Rocky Mountain Labs, as I'll say later. The NHP study, we could not get enough non-human primates to do the comparison with the homotypic vaccination. So they weren't available. And so what we did was just the Mosaic 8 vaccination followed by then, the same sorts of assays, but then they did challenges with SARS-2 Delta, which is actually mismatched because what was on the nanoparticle was beta. And then they did a greater mismatch against SARS-1. And then finally, as I'll talk about later, we did, in collaboration with Jesse Bloom's lab, we did deep mutational scanning to map the polyclonal immune responses. Okay, I'm not going to talk about the NHP study in great detail, except to say that what we were vaccinating with, here's the vaccination schedule up here, and then you can see when they did challenges. So the viral challenge was done at the time of this blue bar, and then they took samples of nasal swabs and bronchoalveolar lavage, and that's all in the paper, and there's histology and stuff like that. But I'll just say very quickly that from looking at the binding responses of the antibodies in the serum that we got, we are looking at day 28, day 36, and then day 106. And blue means mismatched and gray means matched. The point is, you can't really, if you look over here at the legend, you can see what's matched and what's mismatched, but we're not seeing any difference in terms of the antibodies that are raised, which means there's a huge amount of cross-reactivity here. And this was very encouraging to us, especially because we were looking at those class 3 viruses, Sarbecoviruses such as Costa-2, and you really can't tell the difference here. So the Mosaic is really making the type of cross-reactive binding responses we wanted. And then we looked at neutralization, both pseudovirus neutralizations that were done in our lab and authentic virus neutralizations that were done at Rocky Mountain National Labs. These are the people, at BioQual. These are the people at BioQual who did the authentic virus neutralizations and the NHP inoculations and challenges. And so I think what they had to look at were variants of concern of SARS-2, and you're not seeing much of a difference in terms of the neutralization activities. And then we did a bit more strains here, and we were encouraged to see responses against Costa-2 and also against the Omicrons. Okay, so what I want to look at now is the assay that the work we were able to do a more thorough investigation, and this was done in human ACE2 transgenic mice. So this is a mouse model that was developed a while ago, and the human ACE2 is driven under a K18 promoter, and these mice, when they're infected with either with these coronaviruses, they get extremely sick and they die, they lose weight and die. And so it's a much more stringent model of infection than a non-human primate model because non-human primates don't die of SARS-CoV-2 infection or SARS-1 infection. And fortunately, most people don't die either. So this mouse model, it's because of the ubiquitous expression of the ACE2, but they actually, it's a very stringent test. And Vincent Munster at the Rocky Mountain Labs recommended that we do this because you can really tell the difference between what we wanted to see is what happens with a challenge after a Mosaic 8 vaccination versus a homotypic vaccination, because most of the new vaccines that were being tested and being published at the time are various types of protein nanoparticle vaccines that are homotypic. And so we wanted to see how well a homotypic would do compared to our Mosaic 8. Okay, so basically here's the experiment. You prime and one boost now, and then you challenge with either SARS-2, the beta version, or with SARS-1. SARS-2 beta is matched and SARS-1 is mismatched. Okay, so once again, you're seeing the schedule here. And so what I'm showing you first are the average amount of weight loss post-challenge. So after the challenge, the mice were necropsied, and most of them had to be sacrificed if they lost too much weight. And then they looked for survival out to actually I think 21 days after the challenge. Okay, so these are the individual mice in each group, and you see the dotted lines are the amount of their weights for individual mice, and then the average in each group are the solid line. So what you can see for SARS-2 beta challenge is that the animals that didn't have a weight loss problem at all were animals that had been vaccinated with either Mosaic 8 or the homotypic. And these are the controls right here. They were vaccinated with just the nanoparticle, so a naked nanoparticle. And the reason they recover right here is that it's not that many animals because most of them had to be sacrificed at this point. And you can see here that from the survival curve that all the animals in the red and the blue groups, so Mosaic and homotypic, survived, and all but one of six, it was one of six, only one of six survived in the control experiment. Okay, so the interesting thing about this is that, remember on the Mosaic nanoparticle, there's only 1/8 as many RBDs on the nanoparticle of SARS-2 RBDs, that is, as compared to the homotypic. And yet the Mosaic worked just as well as the homotypic in this very stringent animal model, animal challenge model. So that suggests that it would work right now as a SARS-2 vaccine, even though it has 1/8 as many of the SARS-2 RBDs. Okay, so the interesting thing was what happened with the SARS-1 challenge. So remember, that's mismatched. So what you can see here is that the group that did not suffer weight loss after a SARS-1 challenge were the Mosaic 8 immunized mice, but the homotypic animals rapidly lost weight, almost as fast as the control animals in black here. And so if you look at the survival curve, what you can see is 100% of the animals challenged with SARS-1 survived in the Mosaic 8 group, but in the homotypic group, they died, not as fast as the control group, but all but one ended up dying. So this is very interesting because it suggests that the Mosaic 8, if we had a vaccine right now that we could use as a booster, and if it was a Mosaic 8 and then we got some kind of spillover, or we got, another variant of concern, if we were using the Mosaic 8 nanoparticle, we'd have more of a chance of protection than we would if we used a homotypic, say, Omicron presenting particle or something like that. Okay, so we've gone on to do other studies, and in particular with the Mosaic 8 nanoparticles. And in particular, we think this is a really great way to raise monoclonal antibodies that could potentially be therapeutics. And so to show proof of principle of this, we collaborated with John Williams and Miso Park at the City of Hope, because they have a beacon instrument that can be used to rapidly isolate single B cells that secrete IgGs that bind to more than one RBD. So what we did was to immunize mice with either Mosaic 8 nanoparticles or with homotypic nanoparticles. Miso isolated monoclonal antibodies. We expressed them and we did binding and neutralization. And then Cheng Fan, a postdoc in my lab, solved a bunch of cryo-EM structures that I'll show you, and then I'll get to this part right now in a minute. Okay, so the monoclonal antibodies that were isolated from mice immunized with Mosaic 8 RBD nanoparticles showed really nice cross-reactive binding. So we only got 5 of them that cross-reacted according to the criteria we used, but they, in green, are the RBDs that were actually on the particles, so those are matched. But they showed binding to mismatched ones also. The antibodies we got from the homotypic immunized mice, because we were using a criterion that they must be cross-reactive, we couldn't get very many. And these are just other things that are reported in Cheng's paper, and it's actually manuscript accepted. It's actually published now. Okay, so if you look now at neutralization, you can see that for the 5 monoclonal antibodies we got from Mosaic 8 immunized mice that showed cross-reactive binding, they also neutralized for the most part, which was very encouraging. And then the monoclonals isolated from the homotypic animals, because we insisted that they show cross-reactive binding, we got really weakly neutralizing antibodies, weakly or non-neutralizing antibodies. And that's, if we'd selected for antibodies that bind to the more exposed strain-specific epitopes, we would have gotten better ones, but it was the way we designed the experiment. Okay, I just want to show you quickly some of the structures. This particular antibody here, M8A31, was nicely cross-reactive for neutralization across the strains. And Cheng did structures of all of these by cryo-EM. So these are, again, spike trimers, and then the variable domains are from the antibody. This antibody M8A31 is shown, and she solved this bound to the original Washington 1 strain of spike here, and then she also solved it bound to Omicron BA.1 spike trimer, and you're not going to see any difference here. This is a close-up of the interaction with the isolated RBD, and you really can't tell the difference, and it recognizes both the same way. And then very quickly, here are her 5 structures from, these are all Washington 1 spikes, and these are the 5 monoclonal antibodies mapped with their complementarity determining loops shown on the RBD surface. Their binding footprints, heavy chain is always darker than the light chain on the RBD. And then you can see that 4 of these bind to pretty much the same place on the RBD. The 5th one binds to a different place. And this is the really encouraging thing to us was these antibodies, these 4 right here, their binding epitopes are shown in this dotted green line, and they're mostly on this purple region here. So just like we wanted, we're getting antibodies that mainly recognize the purple region, and these we would classify using our nomenclature. 4 of the 5 were class 1/4, and the 5th one was a class 3, and it's actually very similar to 2 of the class 3 antibodies. One of the components of Evusheld was a class 3, and also Sotrovimab. So we raised things that are pretty much comparable to what were being used until very recently, and these don't work as well against Omicrons, and you can see why. It's because they map more to the orange region. Okay, so what we're able to do with all those structures is answer this question, because we've been assuming that actually you can get something like this going on. Like we've been saying, if you have a a membrane-bound B cell receptor, it can reach between adjacent purple regions to cross-link. And so is that really true? Does this architecture support that? So to answer that, what Cheng did was to make models of intact IgGs. So her cryo-EM structures are just the Fabs bound, but you can then model those into what an IgG would look like. And remember, there's flexibility between Fabs. If you have an FC region and you have two Fabs of an IgG, there's flexibility. And so she modeled them with the right, the right aspect ratio here. The sizes are comparable. And these are the real structures of how the IgG would bind to adjacent to the epitopes. For each of the monoclonals, it's possible to cross-link if you have an IgG. So that's representing a membrane-bound B cell receptor, and you could do more here. There could be another one back here, and there could be 3 or 4 or 5 or more of these. So this idea up here appears to be consistent with the modeling. Okay, but of course, that's individual monoclonal antibodies, but the immune response is polyclonal, not monoclonal. So to address that, we collaborated with Jesse Bloom and specifically Ali Greeny, then an MD PhD student in his lab, to do what they've set up as deep mutational scanning. So what they do is they introduce mutations into all 200 positions within a receptor binding domain. And then they evaluate binding of either monoclonal antibodies or polyclonal antisera to a yeast RBD library to identify epitopes, like what changes affect binding. So you've got, close to 4,000 mutations. Not all of them fold properly, but the ones that fold properly are represented by individual yeast here. And so the experiment was to prime and boost with either Mosaic 8 or with homotypic, and then do the mapping. So this was really very satisfying to us. What it showed was that the Mosaic 8, the antisera maps differently than the homotypic antisera. And so the homotypic antisera is down here. What that does is the binding maps to sites in the class 1 and class 2 region. And this is just what you get if you map antisera from vaccinated people or people infected with COVID, is you mainly get antibodies to these class 1 and class 2 regions. But you can see we've shifted it over to this region here, which is actually mostly class 4 or class 1/4. So that's what we were aiming for. And from the deep mutational scanning, that's what we got. So we were very happy about that. So these are mapping mostly to class 4 and class 1/4, and the homotypic is mapping mostly to class 1 and class 2. Okay, so now what people always ask me is, okay, that's great. You're probably, you've shown some evidence that you're preferentially eliciting antibodies to these purple regions, but if you did a vaccine, probably you'd drive mutation of the purple regions. And here's where I'm going to get back to what I introduced at the beginning, which is that there's something really weird to me anyway about the fact that the coronavirus spike trimers exhibit their receptor binding domains most of the time in a down position, but they have to go into an up position in order to bind their receptors. And so if you look at the conserved regions, this is one of the RBDs highlighted here, and the conserved regions are these purple ones. If you now take the RBD the way we've been showing it to you, and you map purple and orange and this gradient across it, these are 2 different views of the RBD. And so this is the class 1/4 region we're trying to target. And if you look at that, now you look at where are the regions of the RBD that contact the rest of the spike trimer when the RBD is down. Because part of the life cycle is that the RBDs must be down at least part of the time on these spike trimers, and so they contact the rest of the spike. And so here's the places that are involved in contacts in blue, and note that from both sides, the blue regions correspond very similarly to the purple regions down here, So I think there's a structural reason that across animal Sarbecoviruses and across the variants of concern, we're really not seeing that much variability in these purple regions. It's a structural reason. And so perhaps what I said here was, you know, these purple regions should, in quotes, remain conserved. And I don't, you know, this virus keeps surprising people, but I think that there's a real potential thing to be glad about, which is that if we can target these purple regions here, I think we might be able to make a vaccine that would not need to be updated for new variants and would not need to be updated for new Sarbecovirus spillovers. Okay, so I wanted to take the next 5 to 10 minutes, maybe a little bit more, to describe something else we're working on. So in this sense, I want to shift gears to talk about a different project that's still coronavirus focused, but it's, for this, I want to think about how mRNA vaccines work, as you'll see why in a minute. Okay, so most of us have gotten some form of an mRNA vaccine. And the interesting thing about them is that they, you know, you take a messenger RNA that's been modified to not trigger inflammatory events, and you put it in a lipid nanoparticle and you deliver it to a cell. The mRNA is translated, and then spike trimers go to the cell surface where they activate B cells. So you make antibodies, and most of the neutralizing antibodies are against the RBD, but you also activate T cells, cytotoxic T cells in particular, because peptides that are translated from the spikes are presented on class 1 MHC molecules. And so this brings me back to, you know, my first introduction into science, which was structural studies of class 1 MHC molecules that are presenting peptides. And so a lot of the reason that the mRNA vaccines still work for us in the face of a lot of spike trimer variation in the RBDs is that there are conserved peptides encoded throughout the spikes that can be presented as short, like 7 to 10 residue peptides presented on MHC class 1 molecules, and you can activate cytotoxic T cells to kill virally infected cells. Okay, so that's great. But in a natural infection, you actually also have viruses that trigger B cell responses. But in an mRNA vaccine, you don't have anything like a virus or a virus-like particle. So the B cell activation is probably not optimal, because if you also had these, it would be better. So what I'm going to tell you about is a technology that Magnus Hoffman invented in my lab to actually do both in an mRNA vaccine. So it's everything like a Pfizer or Moderna vaccine, but it also secretes virus-like particles that could go around and trigger more B cell activation. So the question is, what if you could more closely mimic a natural viral infection by making an mRNA vaccine that expressed spike trimers at the cell surface, just like what we have now, but also secreted virus-like particles with spike trimers on them? So to do this, he took advantage of the escort system, which means endosomal sorting complex required for transport. So this is from a review about escort proteins, and they're doing all kinds of stuff in our cells. They're responsible for their machinery in cells that allow exosomes to be secreted and various types of vesicles. And enveloped viruses like HIV use the escort system. You can see it symbolized here by this blue ring to bud out of virally infected cells. And they're used during cell division as well to separate cells. And so they're also used to make the particles inside multivesicular bodies, and many other things involve escorts. So it's a host cell system that has a lot of components to it. So what Magnus thought about when he was a graduate student is he's now got an NIH Early Independence Award. So he's running his own lab at Caltech. But so when he was a graduate student, he started thinking that he could harness the escort pathway to actually add something to the cytoplasmic tail of, say, a spike trimer that would recruit these escort proteins, and then you'd make these vesicles here that are virus-like particles with membrane-bound spike. Okay, so this is now when he makes those virus-like particles, we call them spike EABR. It means escort and Alex binding region. That's what he added. These are virus-like particles, and they get secreted from transfected cells. And this is cryo-electron tomography done by Ji Yang in my lab, who is now a postdoc at Berkeley, but he did this cryo-ET. So we can look at these, and if you watch this movie enough, you'll see on the grid, these are purified from cell supernatants with a sucrose gradient. You can see like these densely coated particles here. We did a bunch of analyses, and very few of them are bald, meaning almost all of them have spikes. This is a close-up of one of them. It's probably 40 to 60 nanometers. These vary in size, but about that. And you can see this is a slice of a tomographic reconstruction, and that on the right is the model of typically what they would look like, and they have about as many spikes on them as a real SARS-CoV-2 virus. And because they're smaller, a real SARS-CoV-2 virus is 100 to 140 nanometers. These are 40 to 60, same number of spikes, so that means they're more densely coated. Okay, so this is now, we're calling it a hybrid mRNA vaccine, and it encodes self-assembling virus-like particles. And we've shown in this bioRxiv preprint that's out now that actually this works if you do it as an mRNA. So what Magnus did was he took mRNA encoding spike, he modified it with 200 amino acids on its cytoplasmic tail. So it's got, you can read more about it, but it's got a couple of different components here added to the cytoplasmic tail. And now when you do this, you translate it, you go to the cell surface, we've shown that that happens, and you secrete these VLPs. We've shown that that happens if you deliver an mRNA in a lipid nanoparticle. And so he's done this now and shown it works by escort recruitment by various experiments that are in the paper. He's shown it that escort recruitment works to make these secreted virus-like particles for coronavirus spikes, for HIV envelope trimers, for influenza hemagglutinin. You can also make, if you want it to, vesicles that secrete G protein coupled receptors. And probably this is going to work for pretty much anything you try. And in the preprint, we described that if you make mRNA and enclose it in a lipid nanoparticle, which we'll call SEABR, compared to S, which would be the conventional Pfizer or Moderna vaccine, you get higher neutralizing titers compared to the conventional vaccines. And we get increased neutralizing titers against Delta and Omicron variants of concern. And so we think that these higher neutralizing titers could lead to more durable protection. Okay, so with that, I'd like to thank the people in my lab who've done this. And I should say that going back to the first part with the Mosaic nanoparticles, this is being funded by the Coalition for Epidemic Preparedness Innovations. It's funding a clinical trial to evaluate a Mosaic 8 nanoparticle vaccine in humans. So we're working on regulatory stuff to get that going. Okay, so then I started with COVID related structural studies. Christopher Barnes started that at the beginning of the pandemic. He's now an assistant professor at Stanford. So I'm sure he's looking for great postdocs if there's any graduate students who are interested in applying to a really good lab. Cheng Fan is a relatively new postdoctoral fellow in my lab that's, now she's probably solved 40 or 50 cryo-EM structures of coronavirus spike trimers as well. I haven't had a chance to talk about what the other people did in great detail. Originally, we were working on neutralizing antibodies from Michel Nussenswag's lab, from various people in his lab. And then the vaccine design, the Mosaic 8 nanoparticles, is Alex Cohen, who's now a postdoc in my lab. Jennifer Keefe is a staff, this is her, a staff scientist who's running all aspects at Caltech for the clinical trial that we're trying to do. And Magnus is now the person who has a NIH Career Independence Award, who invented the EABR technology. At Oxford, we're collaborating with Alan Townsend, Jack Tan in his lab, Mark Howarth, the Spycatcher Spytag people. At Rocky Mountain National Labs, Vincent and Neilcha for the the ACE2 transgenic challenge studies. At BioQual for the NHP. At City of Hope, John and Miso for the monoclonal antibody, and at the Fred Hutchinson, Jesse, Ali, and Tyler Starr for doing the deep mutational scanning. So I'll stop now and take any questions.
Thank you, Pamela. It's beautiful. People can put their hand up in the chat box, but let me start with a question for you, Pamela. This is really fascinating. It's very cool. A couple questions. One, do you think you showed a primary immunization with your Mosaic and then a boost following that primary, right? Do you think there would be any differences if, like in a clinical trial, the subject was primed with the standard RNA vaccine and then boosted with a Mosaic?
Yeah, see, that's the $1,000,000 question, because we were actually in the middle of that experiment, in animals anyway. So the animals that I showed you the data for were immunologically naive, but obviously people, almost no one is these days. And so the clinical trial will be done in people who've already been exposed, and we want to make absolutely sure that we still get these antibodies against the conserved region. And so what we've got is a series of animals that have been, well, we couldn't get any real mRNA vaccines for a long time, which was frustrating. But anyway, so we first started with AstraZeneca. We were able to get that from Oxford, and we started a bunch of animals 140 days ago that were primed and boosted like people would be. And we waited a while to simulate, you know, that maybe you were vaccinated a while ago, and now those have been injected with Mosaic 8. So we don't know yet. And then more recently, we got the equivalent of a Pfizer vaccine that was formulated. It was modified mRNA formulated in the LNPs, and we started the same experiment in mice. And so we're repeating everything. We'll do the deep mutational scanning. And so that's a really great question. We just don't know based on animal studies yet.
I'm glad you're addressing. That's pretty good. There's a question in the chat. Let me read that to you. It's from Nicole Tobin. She says, fantastic talk. Thank you so much. She always learns a lot. Is there an optimal amount of spike trimers to be expressed on a cell surface to maximize the immune response?
Well, I don't know. Remember, our Mosaic nanoparticle is presenting RBDs, not spike trimers. And, oh, you mean in an mRNA vaccine? Is that...
Nicole, do you want to chime in what exactly you meant?
Yeah, I am curious, and I'm curious if we know the amounts that are expressed and how much it varies between individuals. I haven't seen anything like that.
Neither have I, and I've directly asked people from Moderna and Pfizer when they're at a meeting I'm at where they're giving a talk, and I honestly think they don't know.
Right. I think part of my question comes from adolescents, as we know, have kind of a maximal response with the current doses of those vaccines and a higher risk of adverse events from the vaccinations. And so I'm wondering if we could identify kind of an optimal number that would give you your best immunogenicity and decrease any potential adverse events.
But the adverse events are probably something to do with the adjuvanting effects of the LNPs. You know, they're different from Moderna to Pfizer. The LNPs are all proprietary and they're somewhat different. And they had to, you know, they have interesting immune effects on their own, which are independently of expressing the spike trimer. And so if I were to guess, I would think the adverse effects are not due to expressing spike trimers. They're due to VLNPs. But I mean, we don't know, one of the things we want to do is use this EABR technology to make very brightly labeled spike trimers that are fluorescently labeled, and then look in an animal model to see which cells express them and then where the VLPs go, because that really interests me. Like if you're talking about, this kind of thing happening where the VLPs are released, there's going to be spike trimers here, and I don't know how clustered they are. Like, there's one thing about how many there are on a cell, but it's also about whether or not they're clustered, because that would affect antibody binding.
Right.
So I don't think anybody knows that. Maybe they cluster into subdomain, or lipid microenvironments. Don't know if that's known. But then once you release this, where does it go? And how far does it travel? And what are the immune responses elicited by this compared to that? And so we can do an experiment where we inject just these, we purify these and we inject just those. Then we do a mRNA experiment where we have both of these going on, and then we compare that to the conventional vaccines, and we look at where everything is and what the immune responses are. And so there's a lot of kind of really interesting basic immunology here that we really want to look at. Yeah, I should say I forgot to say this, but I think it's in the bioarchive preprint. Yeah, I think so. We did ELISpot assays, so we do get T cell responses, just like you would if it was a conventional mRNA vaccine. I forgot to say that. But I'm interested in, like, what are the differences in the, are there different types of antibodies elicited if we make these VLPs? Like, do they do different effector functions? There's just kind of an endless number of questions here.
They are really fascinating. Thank you.
Pamela, can I ask you a quick question? You had talked about other related viruses. Do you expect that your Mosaic approach would have any effect on common cold viruses?
No, it doesn't. It doesn't even come close to that. It doesn't even do MERS. That's a beta coronavirus. It does not do merbicoviruses. So we made a Mosaic 8 merbico Mosaic, and are working on that, because we think it would have to be separate. If you want to make a thing on common cold viruses, people are trying to get antibodies to the stalk region, which, so there are papers about you can make antibodies to say the fusion peptide region and places on S2 that are more conserved. There are pictures of this where S2 is in gray, dark gray. Yeah. So this is much more conserved than S1. And so I think if you want to get a pan coronavirus vaccine, which I think is going to be very hard to get, but people are trying, you'd have to target this region, which there are approaches to do that. You can think about it. When I think about that, I don't think it's going to work, because all of us have antibodies raised many times against common cold coronaviruses, right? But we still got this pandemic of SARS-CoV-2. So whatever antibodies we had against years of common cold viruses didn't help us that much.
Yeah. All right. Thank you. We're just about out of time. Any other questions? All right. Well, Pamela, thank you very much. That was a fantastic talk. We really appreciate it.
Okay. Thank you.
Thank you. Everybody enjoy the rest of your day.
Thanks. Bye.
Bye.